to Biology 121 Online. This is the 9th video in our series, and this video is about DNA expression, the first part of DNA expression, DNA to RNA transcription. Before we move on, just a word about expression. As we've said before, the idea about DNA expression is that not every piece of DNA that you have in your entire genome will ever do anything. Some of it doesn't ever do anything, and we'll talk about why that is today. But the parts of your DNA that do get used, we call that expression of that DNA, and we'll talk about how that usage happens. So first of all, recall that a gene is a recipe for how to make a protein. That's what your DNA does. Your DNA tells your body how to make the proteins that form all of your biological functions. So where is DNA located within your cells? Does it ever move? And where are proteins produced within your cells? So ponder that for a moment. And in terms of the answer, does DNA ever move? Well, yes, but only during cellular reproduction. As we said yesterday, part of mitosis involves dissolving the nuclear membrane and splitting sistered chromatids into two parts. But that's not a functional time for the cell. While the cell is reproducing, it's not actually doing anything else. So when the DNA is being expressed is in a different part of the cell cycle. That's during that gap 1 phase. So, taking a look at an animal cell, here we have our nucleolus. Remember that is the actual chromosomes bound together and squished together down into inside this nuclear envelope. So this nucleus protects the DNA itself from things like harmful UV radiation. And that's where the DNA lives, but the proteins are assembled outside of the nucleus at these ribosome sites, many of which are actually attached to the nucleus or to the rough endoplasmic reticulum. So the question is, how does the recipe for making a protein get from inside the nucleus out to where the ribosomes are going to assemble that protein? So that's the first part of a two-step process for building a protein. First, the nuclear DNA that recipe needs to be copied onto a template that can leave the nucleus. Think of it like a library where there are some books that you're not allowed to check out. Well, if you're gonna get access to that information, if you want to take the information outside the library, you're allowed to do that. You just can't take the book. So what do? You make a copy of it. You write down what the book said. That's transcription. That's literally what it means to transcribe something, is to write it down. Then, once that transcribed recipe leaves the nucleus, it'll be assembled as a sequence of amino acids at a ribosome, and we call that process translation, and that's going to be a separate video on Monday. So, let's talk about transcription specifically. The DNA inside the nucleus is engulfed by an enzyme. This is how this works. So there's an enzyme called RNA polymerase, and like most enzymes, it's named for exactly what it does. It polymerizes or constructs a long chain of RNA. So ribonucleic acid, very similar to deoxyribonucleic acid. The big difference is ribonucleic acid's spin rate as it coils around in a spring-like structure doesn't allow for the double helix of DNA, and also that spin rate doesn't allow the thymine base to participate in the RNA. So instead of thymine, RNA uses uracil. So instead of T's, RNA uses U's. More on that later. So once we have, I should note, the DNA that's being copied is only one of the two strands of the DNA double helix. So we call it a template strand because it's the side that has the actual encoding that's going to create the right sequence of amino acids for the protein. The other side from the template of the DNA is just called the complement. So, metaphorically, this long strand of RNA, once it's been created as a complement to the template DNA strand, is called a messenger and is labeled as mRNA. Now, I flagged a question. Why do we call it a messenger? Well, imagine that library, again, that won't let you take out a book. If you can't go into the library yourself to make a copy of the book, what do? Well, you send a messenger to go get that information for you, right? So for that reason, we call mRNA, messenger RNA, the recipe for the protein copied from a template. So here's a visualization of that. You can see this DNA double helix, and this DNA double helix is not shown spun just for clarity. It really would be twisted throughout the entire left-hand side and the right-hand side, right? So these should look twisty, but they've just simplified it to make it look like a ladder. And then the RNA polymerase is opening up that DNA double helix, and this template strand is being copied so that really, if this is like guanine here, then that's going to be a cytosine. If this is an adenine, then that's going to be a uracil because it's replacing thymine. If this is a thymine, that's going to be an adenine. So the RNA, messenger RNA strand, ends up matching not the template strand, but it ends up being exactly the same as the complement, the coding strand. So the template strand is the complementary match to this coding strand, and the RNA is the same as the coding strand. So it's kind of like a double negative, right? If we look at the bottom of the page here, on the coding strand, we have an adenine, a thymine, a guanine, an adenine, a thymine, a cytosine, a thymine. And on the template strand, adenine goes with thymine, thymine goes with adenine, guanine goes with cytosine, adenine goes with thymine. But then the RNA matching against the template strand ends up being the same as the coding strand, with the exception that thymine in DNA is replaced with uracil in RNA. So, some of this DNA that's on that coding strand isn't actually needed for the protein recipe. We used to call these stretches junk DNA. If you've ever heard the phrase junk DNA, that's what they're talking about, the parts of your DNA that don't actually produce a protein. We now call those parts introns because we have a much better understanding of why they're there. After the RNA is produced, the introns are removed, and the remaining mRNA, which we call exons, are spliced together and reconnected. Now we're going to talk about why these introns are there in a minute, but first let's just look at this process itself. So here we have on the top line the DNA, so that black line down the middle here is meant to convey to you that this is a stretch of DNA. And then they've labeled this stretch of DNA, this sequence of DNA in particular, as exon 1. And there's exon 2, and exon 3, and exon 4. And then the parts where they didn't box it out, those are called the introns. That's what they're labeling as the introns. So this is the DNA that's, the introns represent the DNA that's not going to become part of the protein recipe. And the exons refer to the parts that are going to be part of the protein recipe. So the pre-mRNA is the copy, or the complementary copy of the DNA transcribed as RNA with the introns still attached. So the RNA polymerase does that job of creating this copy. And you'll notice that the pre-mRNA, all of the pieces match up with what the DNA had. It's just a copy. So then there's something called a spliceosome, which is just a body that does some splicing. And that spliceosome is going to remove these introns, leaving just the exons directly connected to each other. So let's look at that process of what happens during splicing. So here we have a precursor mRNA molecule. So this is our pre-mRNA. In blue, we have the exons, and in yellow, we have an intron. So at the beginning of that intron, there's a splice site, and near the end of that intron, but not at it, there's an attachment site for another protein that's going to help out with this process. So these two little sort of beige tan balls represent two different proteins that bind to the RNA and then pull together, as you can see down here. And so the exons are brought closer to each other as these two proteins get attracted towards each other, and then other proteins also come in and become involved. One of those proteins actually cuts the RNA molecule. Another protein takes the loose end of the RNA molecule and attaches it to the site where the second protein here had bound. And then other proteins cut this strand of RNA right there at the splice site. And yet another protein is going to take this exposed end and then on the left-hand side exon and this exposed end on the right-hand side exon and splice those two together, while the lariat, they call it because it looks kind of like a lasso, this lariat of intron RNA is going to be let loose within the nucleus, and it's just going to like fall back apart into loose nucleotides ready to be reused again in the next expression of DNA. So in that way, we kind of grab the piece of RNA, pull it together, and cut it out. Now, there's a specific DNA sequence. You might ask, like, how does it know where to cut? And the answer is that there are specific DNA sequences, both that tell RNA polymerase where to start making the pre-RNA, that's called the promoter site, and here it says the specific sequence in mammals is TAA. So there's a thymine and two adenosines, and that tells, that's always present whenever there's a promoter site within a DNA sequence. There's other sequences that tell the RNA ligase where to cut and where to splice, and they are called lariat sequences. And you can see again, it's a lot of TAAs, TGAs, and TAGs. And we'll talk about why those ones, why those specific sequences are instructions to the helper molecules, the helper enzymes, RNA polymerase and RNA ligase, and all four of those other blobs that were on the last picture, to tell those proteins where to begin and end this DNA sequencing. So why are there introns? You remember the last video, we talked about how DNA in its double helix gets wrapped around a histone, and then those histones get kind of bundled together in such a way that some of this DNA is readily accessible and can be read and can be interacted with by proteins that are inside the nucleus. And other stretches of the DNA, like where my pointer is now, are down deep inside this structure of the chromosome, where proteins won't really be able to access and interact with those sequences of DNA. So one of the functions that we know introns perform is that they ensure that the promoter sequence of a gene is exposed on the chromosome so that proteins like RNA polymerase can find it and know that the gene starts there. One of the other things that has been hypothesized about introns is that in some cases, a single stretch of DNA may actually have sequences of more than one particular gene. So there may be different recipes or parts of different recipes superimposed in upon each other. And if we take that recipe metaphor all the way, you might consider there's lots of recipes that call for 3 eggs. So rather than write down on every page of the recipe book, this recipe has 3 eggs, DNA in some cases appears to actually have a sequence that specifies a particular stretch of amino acids, a particular sequence of amino acids, and it'll use that same instruction in multiple different protein recipes. So, just to review, once the mature mRNA, so this is our mature mRNA down at the bottom, having started as a DNA sequence, being converted into an RNA sequence, and then having the introns cut out and becoming a mature strand of messenger RNA, once those exons are removed, it is ready to leave the nucleus, go find a ribosome, and get the process of creating a protein started. But that's going to be what we talk about in our next video.